Picture yourself standing at the edge of something so vast, it almost defies comprehension. Not a canyon or an ocean, but something far more intimate. Something that is you. Inside your body, at this very moment, there are approximately 37 trillion cells. 37 trillion individual living units, each one a microscopic universe unto itself, all working in perfect concert to create the person experiencing these words right now. Welcome to Bedtime Biology, where science meets serenity. Each night, we explore the quiet wonders of the natural world, calming your mind and sparking your curiosity. Tonight, we're embarking on a journey through one of nature's most extraordinary stories. We'll travel from a single cell, smaller than the period at the end of this sentence, all the way to the complex, breathing, thinking being that is you. We'll discover how cells, those microscopic building blocks of life, communicate with one another, specialize into hundreds of different forms, and organize themselves into the tissues, organs, and systems that allow you to move, think, feel, and simply be alive. This is a story that begins before you drew your first breath, before you had a heartbeat, before you even had a heart. It begins with a moment so quiet and so small that it would be invisible to the naked eye, yet so monumentally important that everything you are stems from that single instant. Let's slow down together. Let's explore the hidden architecture of life itself. And let's discover how cells build the human body. The journey of every human being begins in a place of profound simplicity. A single cell, just one. In that moment, when a sperm cell, carrying 23 chromosomes, meets an egg cell, also carrying 23 chromosomes, something remarkable happens. They fuse together in a process called fertilization, creating what scientists call a zygote. This zygote is the very first cell of what will, 9 months later, emerge as a newborn human baby. Think about that for a moment. Everything you are, everything you've ever experienced, every thought you've had, every movement you've made, traces its lineage back to that single cell. That zygote contained within it the potential for all the 37 trillion cells that would eventually form your body. It held the blueprint for your eyes and your fingernails, your brain cells and your blood cells, your skin and your bones. All of it encoded in a single cell so small, you would need a microscope to see it. The zygote wastes no time. Within hours of its formation, it begins to divide. 1 cell becomes 2, 2 become 4, 4 become 8. This process, called cleavage, continues in a carefully choreographed sequence. What's fascinating is that during these early divisions, the cells don't grow between each split. The original egg cell was relatively large, as cells go, and these early divisions simply partition that original volume into smaller and smaller compartments. The total mass stays roughly the same, but the number of cells multiplies exponentially. By the time there are 16 cells, the embryo looks like a tiny cluster of soap bubbles, each bubble a complete cell with its own membrane, its own set of genetic instructions, its own molecular machinery. Scientists call this cluster a morula, from the Latin word for mulberry, because of its berry-like appearance. But this is no ordinary berry. This is the dawn of a human life. The cells continue to divide, and something extraordinary begins to happen. The solid ball of cells reorganizes itself into a hollow sphere called a blastocyst. On one side of this sphere, a cluster of cells gathers, forming what's called the inner cell mass. These are the cells that will eventually form the entire baby. The outer layer of cells will become the placenta and other supporting structures. The blastocyst, by now containing about 100 cells, begins its journey down the fallopian tube toward the uterus, where it will implant itself into the uterine wall and begin the next phase of development. At this stage, approximately 5 to 6 days after fertilization, something remarkable is true about these cells. They are pluripotent. This means that each cell in the inner cell mass has the potential to become any type of cell in the human body. These cells haven't yet decided what they'll be. They haven't committed to becoming a brain cell or a heart cell or a skin cell. They exist in a state of pure potential, waiting for the signals that will guide them toward their ultimate destiny. This pluripotency is one of the most remarkable properties of early embryonic cells. Inside each of these cells is a complete copy of the human genome, approximately 3 billion base pairs of DNA organized into 46 chromosomes. This genome contains roughly 20,000 to 25,000 genes, each one a set of instructions for making a specific protein. Every cell in the blastocyst has access to all of these genes, all of these instructions. And yet, as development proceeds, different cells will activate different subsets of these genes, leading them down different paths of specialization. The process by which these initially identical cells become different from one another is called differentiation, and it's one of the fundamental mysteries and marvels of developmental biology. We'll explore it in depth later in our journey tonight, but for now, simply marvel at this fact. From a single fertilized egg cell, every different type of cell in your body emerged. Your neurons, with their long, branching extensions that carry electrical signals. Your muscle cells, packed with protein filaments that can contract and relax. Your red blood cells, shaped like biconcave discs and filled with hemoglobin to carry oxygen. Your bone cells, your fat cells, your skin cells, your immune cells. All of them, every single one, descended from that original zygote. By the end of the first week, the blastocyst has implanted into the uterine wall, and a new phase begins. During the second week, the cells begin to organize themselves into distinct layers. By the third week, 3 primary layers have formed, and these layers have names that you might recognize from biology class: the ectoderm, the mesoderm, and the endoderm. These 3 layers are like the primary colors of cellular development. Just as all the colors you see can be created from combinations of red, blue, and yellow, all the different cell types in your body will arise from these 3 embryonic layers. The ectoderm, the outermost layer, will give rise to your nervous system, including your brain and spinal cord. It will also form your skin, your hair, your nails, and the enamel of your teeth. The mesoderm, the middle layer, will become your muscles, your bones, your circulatory system, and your kidneys. The endoderm, the innermost layer, will form the lining of your digestive system, your lungs, your liver, and your pancreas. This 3-layered structure, established just 3 weeks after conception, sets the stage for all the complexity that will follow. Over the coming weeks and months, these layers will fold, bend, split, and reorganize in intricate ways, guided by chemical signals and genetic programs, sculpting themselves into the recognizable form of a human baby. Organs will appear. Systems will come online. The heart, one of the first organs to function, will begin beating around day 21, just 3 weeks after that initial fertilization event, pumping blood through a cardiovascular system that is still being built. But before we follow the embryo further along its developmental journey, let's pause and zoom in. Let's look more closely at the cells themselves, because to understand how cells build the human body, we first need to understand what a cell actually is, how it's structured, and what gives it the remarkable capabilities that make life possible. Every cell in your body, whether it's a neuron in your brain, or a cell in your liver, or a white blood cell patrolling your bloodstream, shares certain fundamental features. All cells are bounded by a structure called the cell membrane, also known as the plasma membrane. This membrane is not a solid wall, but rather a fluid barrier, a boundary that separates the interior of the cell from the outside world. The cell membrane is made primarily of molecules called phospholipids. Each phospholipid molecule has 2 distinct parts: a head that is hydrophilic, meaning it's attracted to water, and 2 tails that are hydrophobic, meaning they repel water. In the watery environment, both inside and outside the cell, these phospholipids spontaneously arrange themselves into a double layer called a bilayer, with the hydrophilic heads facing outward toward the water on both sides, and the hydrophobic tails tucked away in the middle, shielded from the water. This phospholipid bilayer forms the basic structure of the cell membrane, but it's far from simple. Embedded within this bilayer are thousands of different proteins, each serving specific functions. Some of these proteins act as channels or pores, allowing specific molecules to pass into or out of the cell. Others act as pumps, using energy to move substances against their concentration gradients. Still others serve as receptors, binding to specific molecules outside the cell and transmitting signals to the cell's interior. The cell membrane also contains cholesterol molecules, which help regulate the membrane's fluidity, and various carbohydrates attached to proteins and lipids, forming a sugar coating that helps cells recognize one another. The cell membrane is selective about what it allows to pass through. Small, non-polar molecules like oxygen and carbon dioxide can slip through the phospholipid bilayer relatively easily. So can small uncharged molecules like water, though even water crosses more slowly than you might expect. But larger molecules, charged molecules, and ions cannot simply diffuse through the membrane. They require special transport proteins to help them cross. This selective permeability is crucial. It allows the cell to maintain a different internal environment from the external environment. Inside the cell, the concentrations of various ions, nutrients, and other molecules can be precisely controlled, creating conditions optimized for the chemical reactions that sustain life. Without this selective barrier, cells would quickly equilibrate with their surroundings, losing the gradients and differences that are essential for cellular function. Enclosed by this membrane is the cytoplasm, the jelly-like substance that fills the cell. The cytoplasm is not just a simple liquid, but a complex, crowded environment. Dissolved in the water that makes up most of the cytoplasm are countless molecules: proteins, sugars, amino acids, nucleotides, ions, and many others. The cytoplasm also contains an intricate network of protein filaments called the cytoskeleton, which gives the cell its shape, provides structural support, and creates highways along which materials can be transported within the cell. Suspended within the cytoplasm are specialized structures called organelles. Each organelle is like a tiny organ within the cell, performing specific functions essential to the cell's survival. Let's explore some of the most important ones. The nucleus is perhaps the most prominent organelle in most human cells. It's often described as the control center of the cell, and for good reason. The nucleus houses the cell's DNA, organized into structures called chromosomes. In human cells, there are 46 chromosomes arranged in 23 pairs. 22 of these pairs are called autosomes, and they're essentially the same in males and females. The 23rd pair consists of the sex chromosomes, 2 X chromosomes in females, and 1 X and 1 Y chromosome in males. The nucleus is enclosed by its own double membrane, called the nuclear envelope, which is studded with thousands of tiny pores. These nuclear pores act as gateways, allowing certain molecules to pass between the nucleus and the cytoplasm, while keeping the DNA safely sequestered. This separation is important because it allows the cell to regulate gene expression more precisely, controlling when and how the information in DNA is accessed and used. Inside the nucleus, the DNA is not just floating around randomly. It's wrapped around proteins called histones, forming structures that look like beads on a string. These beads, called nucleosomes, can be packed together more or less tightly, and this packing has important consequences for gene expression. Genes in tightly packed regions are generally inaccessible and inactive, while genes in more loosely packed regions can be accessed by the cellular machinery that reads DNA and makes RNA. This level of organization adds another layer of control over which genes are active in which cells. Also, within the nucleus is a structure called the nucleolus, where a particular type of RNA, called ribosomal RNA, is made, and where the components of ribosomes are assembled. Ribosomes are the cellular machines that translate the information in messenger RNA into proteins, and we'll encounter them again soon. Outside the nucleus, scattered throughout the cytoplasm, are numerous other organelles. Among the most important are the mitochondria, often called the powerhouses of the cell. Mitochondria are unusual organelles in several ways. First, they're surrounded by 2 membranes, an outer membrane and an inner membrane. The inner membrane is highly folded, creating structures called cristae that greatly increase its surface area. Second, mitochondria contain their own small genome, a circular piece of DNA that encodes a handful of genes. This mitochondrial DNA is passed down exclusively from mothers to their children, creating an unbroken maternal lineage stretching back through evolutionary time. The primary function of mitochondria is to produce ATP, adenosine triphosphate, the cell's main energy currency. They do this through a process called oxidative phosphorylation, which we'll explore in greater detail later. For now, know that mitochondria take the breakdown products of the food we eat, the carbohydrates and fats and proteins, and extract energy from them, storing that energy in the high energy bonds of ATP molecules. Every time your cells need energy to do work, whether it's to build new molecules, to transport substances across membranes, to move, or to send signals, they typically break down ATP, releasing that stored energy. Different cells have different numbers of mitochondria, depending on their energy needs. A typical liver cell might have 1,000 to 2,000 mitochondria. A muscle cell, which requires enormous amounts of energy for contraction, might have even more. Heart muscle cells, which must contract continuously throughout your life, can be up to 40% mitochondria by volume. Meanwhile, red blood cells, which don't have nuclei and don't produce their own ATP, have no mitochondria at all. Another crucial organelle is the endoplasmic reticulum, often abbreviated as ER. This is a vast network of interconnected membranes that extends from the nuclear envelope throughout much of the cytoplasm. There are two types of ER, rough ER and smooth ER. Rough ER gets its name from the ribosomes that stud its surface, giving it a bumpy appearance under an electron microscope. These ribosomes are busy making proteins, many of which will be secreted from the cell or embedded in cellular membranes. As the proteins are made, they're threaded through channels in the ER membrane into the interior space, where they can be folded properly and modified. Smooth ER, which lacks ribosomes, has different functions depending on the cell type. In liver cells, smooth ER is involved in detoxifying harmful substances. In muscle cells, a specialized type of smooth ER stores calcium ions and releases them when the muscle needs to contract. In cells that produce steroid hormones, smooth ER is where those hormones are synthesized. Closely associated with the ER is another organelle called the Golgi apparatus, or Golgi complex. You can think of the Golgi as the cell's post office. Proteins made in the ER are packaged into small membrane-bound sacs called vesicles, which bud off from the ER and travel to the Golgi. At the Golgi, proteins are further modified, sorted, and packaged for delivery to their final destinations, whether that's another organelle within the cell membrane, or outside the cell entirely. Cells also contain digestive organelles called lysosomes. These are membrane-bound compartments filled with enzymes capable of breaking down virtually any biological molecule: proteins, carbohydrates, lipids, nucleic acids. Lysosomes serve several functions. They digest materials that the cell has taken in from outside. They recycle worn-out cellular components, and they can even help destroy invading bacteria. The enzymes inside lysosomes are so powerful that if they were released into the cytoplasm, they would digest the cell itself. The lysosomal membrane keeps these dangerous enzymes safely contained. Another type of organelle, called a peroxisome, performs various metabolic functions, including breaking down fatty acids and detoxifying certain harmful substances. Peroxisomes produce hydrogen peroxide as a byproduct of some of their reactions, then quickly break it down using an enzyme called catalase before it can damage the cell. This, then, is the basic architecture of a human cell: a plasma membrane enclosing a cytoplasm filled with organelles, each carrying out specialized functions, all working together to keep the cell alive and functional. But cells don't operate in isolation. The human body contains over 200 different types of cells, each specialized for particular roles, and all of these cells must work together in a coordinated way. To understand how this coordination happens, we need to understand the information system that underlies all of cellular life, the genetic code written in DNA. At the heart of every cell, protected within the nucleus, lies the most precious molecule in biology, deoxyribonucleic acid, or DNA. This molecule is both remarkably simple in its structure and astoundingly complex in its implications. DNA is the instruction manual for building and operating a human being, and every cell in your body contains a complete copy of this manual. The structure of DNA is elegant. It consists of two long strands wound around each other in a double helix, like a twisted ladder. The sides of the ladder are made of alternating sugar molecules, specifically deoxyribose and phosphate groups. The rungs of the ladder are made of pairs of molecules called nucleotide bases. There are 4 types of bases in DNA: adenine, abbreviated A, thymine, abbreviated T, guanine, abbreviated G, and cytosine, abbreviated C. These bases pair up in a specific way, following a rule discovered by the scientists James Watson and Francis Crick when they determined the structure of DNA in 1953. Adenine always pairs with thymine, and guanine always pairs with cytosine. This complementary base pairing is crucial because it means that if you know the sequence of bases on one strand of DNA, you can deduce the sequence on the other strand. This property allows DNA to be copied accurately during cell division. The human genome, the complete set of genetic information in a human cell, consists of approximately 3 billion of these base pairs, distributed across 46 chromosomes. Written out letter by letter, the human genome would fill hundreds of thick books. Yet all of this information is packed into the nucleus of each of your cells, a space measuring only about 6 micrometers across. Within this vast sequence of DNA lie the genes, specific stretches of DNA that contain instructions for making proteins. A gene might be a few hundred base pairs long, or it might be hundreds of thousands of base pairs long. The human genome contains somewhere between 20,000 and 25,000 genes, though the exact number is still debated because defining exactly what counts as a gene turns out to be surprisingly complicated. Here's what's remarkable. Not all of your DNA consists of genes. In fact, only about 1 to 2% of the human genome directly codes for proteins. The rest was once dismissed as junk DNA, but we now know that much of it plays important roles in regulating when and where genes are turned on or off. Some of these non-coding regions contain binding sites for proteins that control gene expression. Others produce RNA molecules that don't code for proteins but have regulatory functions. The genome is not just a list of protein recipes. It's a complex, interwoven set of instructions and controls. Different cells in your body have the same DNA, the same genes, yet a neuron looks and functions completely differently from a muscle cell, which looks and functions differently from a skin cell. How can this be? The answer lies in gene expression. While every cell has a complete genome, only a subset of genes is active in any given cell type. Which genes are active determines the cell's identity and function. A neuron expresses genes for proteins involved in sending and receiving electrical and chemical signals, ion channels, neurotransmitter receptors, proteins that help build synapses. A muscle cell expresses genes for contractile proteins like actin and myosin, which slide past each other to generate force. A pancreatic beta cell expresses genes for insulin, the hormone that regulates blood sugar. Each cell type has a characteristic pattern of gene expression, and this pattern determines what the cell can do. Gene expression is a tightly regulated process. Cells can turn genes on or off in response to signals from their environment, allowing them to adapt to changing conditions. They can fine-tune the level of expression, making more or less of a particular protein as needed. The mechanisms that control gene expression are complex, involving multiple layers of regulation, from the structure of chromatin, the complex of DNA and proteins, to the various proteins that bind to DNA and either promote or inhibit transcription, to the processing and stability of RNA molecules, to the translation of RNA into protein. The path from gene to protein involves 2 major steps: transcription and translation. Let's walk through these processes because they're fundamental to understanding how cells work. Transcription is the first step, the process by which the information in a gene is copied into a molecule of RNA. RNA, or ribonucleic acid, is chemically similar to DNA, but with a few key differences. RNA is usually single-stranded rather than double-stranded. It uses the sugar ribose instead of deoxyribose, and it uses the base uracil, abbreviated U, instead of thymine. Transcription begins when a large enzyme complex called RNA polymerase binds to a region of DNA near the beginning of a gene, a region called a promoter. The RNA polymerase unwinds the DNA double helix, exposing the bases. It then moves along one strand of the DNA, the template strand, reading the sequence of bases and synthesizing a complementary strand of RNA. If the DNA template strand has a T, the RNA polymerase adds an A to the growing RNA strand. If the template has an A, the RNA polymerase adds a U. If the template has a G, the RNA adds a C. And if the template has a C, the RNA adds a G. This process continues until the RNA polymerase reaches a signal in the DNA that tells it to stop, typically a specific sequence of bases called a terminator. The RNA polymerase then releases the newly made RNA molecule and detaches from the DNA. The DNA double helix reforms, and the gene is once again a stable, compact structure. The RNA molecule that's produced, called messenger RNA or mRNA, is a working copy of the gene. In bacteria, this mRNA can immediately be used to make protein. But in eukaryotic cells like ours, there's an additional layer of complexity. The initial RNA transcript, called pre-mRNA, must be processed before it can leave the nucleus and be translated into protein. This processing involves several steps. First, a cap is added to one end of the RNA, and a tail of many adenine bases is added to the other end. These modifications protect the RNA from being degraded and help it to be recognized by the cellular machinery that will translate it. Second, and more remarkably, the pre-mRNA is spliced. Eukaryotic genes are not continuous stretches of protein coding sequence. Instead, they're interrupted by segments called introns, which don't code for protein. The coding segments are called exons. During splicing, the introns are cut out and the exons are joined together, creating a mature mRNA molecule that contains only the protein coding information. This splicing process is carried out by a large complex called the spliceosome, which recognizes specific sequences at the boundaries between introns and exons. Remarkably, a single gene can sometimes be spliced in different ways, including different combinations of exons in the final mRNA, a process called alternative splicing. This allows one gene to encode multiple different proteins, greatly increasing the diversity of proteins that can be made from a limited number of genes. Once the mRNA has been processed, it's transported out of the nucleus through the nuclear pores and into the cytoplasm, where it will be translated into protein. Translation is the process by which the sequence of nucleotides in mRNA is decoded to produce a specific sequence of amino acids, the building blocks of proteins. The key to translation is the genetic code, a set of rules that maps each possible sequence of 3 nucleotides, called a codon, to a specific amino acid. There are 64 possible codons, 4 possible bases in the first position, times 4 in the second, times 4 in the third, equals 64. But there are only 20 standard amino acids used to build proteins. This means that the genetic code is redundant, with most amino acids specified by more than one codon. 3 of the 64 codons, called stop codons, signal the end of the protein coding sequence. translation takes place at ribosomes, large molecular machines made of ribosomal RNA and proteins. Each ribosome has two main parts, a large subunit and a small subunit, which come together around an mRNA molecule to begin translation. The ribosome reads the mRNA 3 nucleotides at a time, 1 codon at a time, moving from one end of the message to the other. But the ribosome doesn't directly recognize the codons. Instead, it relies on molecules called transfer RNAs, or tRNAs. Each tRNA has two important features: a 3 nucleotide sequence called an anticodon, which can base pair with a specific codon in the mRNA, and an amino acid attachment site, where a specific amino acid is covalently bonded to the tRNA. The pairing between codon and anticodon ensures that the correct amino acid is added to the growing protein chain at each step. When a tRNA with the correct anticodon binds to a codon in the ribosome, the amino acid it carries is added to the end of the growing protein chain, forming a peptide bond with the previous amino acid. The ribosome then moves to the next codon, and the process repeats. This continues until the ribosome encounters a stop codon. At that point, no tRNA can bind, because there's no tRNA with an anticodon matching the stop codon. Instead, proteins called release factors bind to the ribosome, triggering the release of the completed protein and the disassembly of the ribosome. The protein that emerges from the ribosome is simply a chain of amino acids. This chain must fold into a specific 3-dimensional shape to become functional. The sequence of amino acids determines the final shape, because different amino acids have different chemical properties. Some are hydrophobic, avoiding water. Some are hydrophilic, attracted to water. Some are positively charged, some negatively charged, some neutral. As the protein folds, these different amino acids interact with each other and with the surrounding water, guiding the protein toward its functional shape. Many proteins also undergo further modifications after translation. They might have chemical groups added to certain amino acids, or they might be cut into smaller pieces, or they might have sugars attached to them. All of these modifications can affect the protein's function, stability, or location within the cell. This process, from DNA to RNA to protein, is sometimes called the central dogma of molecular biology. It represents the fundamental flow of information in living cells. The information stored in DNA is transcribed into RNA, which is then translated into protein. And proteins, in turn, do most of the actual work in cells. They catalyze chemical reactions as enzymes. They provide structural support. They transport molecules. They send and receive signals. They regulate gene expression. They defend against pathogens. In a very real sense, you are your proteins. Your physical form, your metabolism, your ability to think and move and sense the world, all depend on the thousands of different proteins your cells make. But making all these proteins requires energy, and lots of it. Translation alone consumes a substantial fraction of a cell's energy budget. Cells need a constant supply of ATP to power not just protein synthesis, but all the myriad other processes that keep them alive. Transporting molecules across membranes, maintaining ion gradients, building and breaking down complex molecules, moving, dividing, responding to signals. Where does all this energy come from? For that, we turn to the mitochondria. Inside each mitochondrion, a metabolic dance is unfolding, one that has been perfected over billions of years of evolution. This dance extracts energy from the food we eat and transforms it into ATP, the universal energy currency of life. When you eat a meal, the carbohydrates, fats, and proteins in that food are broken down by your digestive system into smaller molecules, sugars like glucose, fatty acids, and amino acids. These molecules are absorbed into your bloodstream and distributed to cells throughout your body. Once inside a cell, these fuel molecules are processed in stages to extract their energy. The first stage of glucose metabolism, called glycolysis, takes place in the cytoplasm. In a series of 10 chemical reactions, one molecule of glucose, a 6-carbon sugar, is split into 2 molecules of pyruvate, a 3-carbon compound. This process doesn't require oxygen, and it generates a small amount of ATP directly. 2 molecules of ATP per glucose molecule. It also generates 2 molecules of NADH, a carrier of high-energy electrons that will be important later. Glycolysis represents an ancient metabolic pathway, one that likely evolved in the early days of life on Earth, before oxygen was abundant in the atmosphere. For organisms that live in oxygen-poor environments, or for cells that occasionally run low on oxygen, glycolysis alone can provide energy. The pyruvate produced by glycolysis can be converted to lactate, regenerating the molecules needed to keep glycolysis running. This is what happens in your muscle cells during intense exercise, when oxygen delivery can't keep up with demand. You feel the burn as lactate accumulates. But when oxygen is available, cells can extract far more energy from glucose. The pyruvate produced by glycolysis is transported into the mitochondria, where it enters a series of reactions called the citric acid cycle, also known as the Krebs cycle, or the tricarboxylic acid cycle. This cycle takes place in the mitochondrial matrix, the space enclosed by the inner mitochondrial membrane. In the citric acid cycle, pyruvate is first converted to a molecule called acetyl-CoA. This 2-carbon acetyl group is then combined with a 4-carbon molecule called oxaloacetate to form citrate, a 6-carbon molecule. Through a series of steps, this citrate is gradually transformed, losing carbon atoms one by one as carbon dioxide and regenerating oxaloacetate, which can combine with another acetyl-CoA to keep the cycle turning. The citric acid cycle doesn't produce much ATP directly. Its main purpose is to generate high-energy electron carriers, NADH, and a related molecule called FADH2. For each acetyl-CoA that enters the cycle, 3 molecules of NADH and 1 molecule of FADH2 are produced. Since each glucose molecule yields 2 pyruvates, and each pyruvate yields 1 acetyl-CoA, the citric acid cycle generates 6 NADH and 2 FADH2 per glucose molecule. These electron carriers are rich in energy because they contain high-energy electrons extracted from the breakdown of glucose. But the cell can't use NADH and FADH2 directly to power most cellular processes. These electrons need to be transferred to oxygen, and the energy released in that transfer needs to be captured in the form of ATP. This is where the real magic of mitochondria happens, in a process called oxidative phosphorylation. The inner mitochondrial membrane contains a series of protein complexes collectively known as the electron transport chain. NADH and FADH2 deliver their electrons to this chain. The electrons then pass from one protein complex to the next, moving downhill in energy with each transfer. At several points in this chain, the energy released is used to pump protons, hydrogen ions, out of the mitochondrial matrix and into the space between the inner and outer mitochondrial membranes. This creates a gradient, a high concentration of protons outside the inner membrane, and a low concentration inside. Protons naturally want to flow back in, driven both by the concentration difference and by an electrical difference, since protons carry a positive charge. But the inner mitochondrial membrane is not freely permeable to protons. They can only flow back through a specific protein complex called ATP synthase. ATP synthase is a molecular machine of extraordinary sophistication. As protons flow through it, driven by the gradient, it rotates like a tiny turbine. This rotation drives a conformational change that brings together ADP and inorganic phosphate, forming ATP. It's a process of breathtaking efficiency, coupling the energy stored in the proton gradient to the synthesis of the high-energy bonds in ATP. At the end of the electron transport chain, the electrons are transferred to molecular oxygen, which combines with protons to form water. This is why we breathe, to provide oxygen as the final electron acceptor in this chain. Without oxygen, the electron transport chain would back up, unable to pass on electrons, and oxidative phosphorylation would grind to a halt. When you add it all up, the complete oxidation of one glucose molecule, from glycolysis through the citric acid cycle to oxidative phosphorylation, can yield about 30 molecules of ATP. This is roughly 15 times more than glycolysis alone produces. This is why aerobic organisms that use oxygen, have such a huge energy advantage and can support complex active lifestyles. Different cells have different energy needs, and the number of mitochondria in a cell reflects those needs. Cells that are metabolically very active, like muscle cells and neurons, contain many mitochondria. Heart muscle cells, which must contract ceaselessly from before birth until death, are packed with mitochondria, which can make up 40% of the cell's volume. Liver cells, which carry out a huge variety of metabolic tasks, also have abundant mitochondria, often more than 1,000 per cell. Mitochondria are not static structures. They constantly undergo fusion, where two mitochondria merge to form one, and fission, where one mitochondrion divides into two. This dynamic behavior allows cells to adjust their mitochondrial networks to match their energy needs and to maintain the health of their mitochondrial population. Damaged mitochondria can be eliminated through a process called mitophagy, a specialized form of autophagy where defective mitochondria are engulfed and broken down by lysosomes. The importance of healthy mitochondria cannot be overstated, because mitochondria have their own DNA and their own protein-making machinery. Mutations in mitochondrial DNA can have serious consequences, leading to a variety of mitochondrial diseases. These diseases often affect tissues with high energy demands, such as muscle and brain, causing symptoms ranging from muscle weakness to seizures to developmental delays. The fact that mitochondrial DNA is inherited exclusively from the mother means that mitochondrial diseases are passed down the maternal line. With a constant supply of ATP from their mitochondria, cells have the energy they need to carry out all their functions. But for a multicellular organism, like a human, to develop, cells must do more than just maintain themselves. They must divide, creating new cells. They must specialize, taking on distinct roles, and they must organize themselves into tissues, organs, and systems. Let's explore how all of this happens. Cell division is one of the most fundamental processes in biology. It's how organisms grow, how they repair damaged tissues, and how they reproduce. In humans and other multicellular organisms, there are two main types of cell division: mitosis, which produces two genetically identical daughter cells, and meiosis, which produces sex cells with half the usual number of chromosomes. Let's begin with mitosis, the type of cell division that's responsible for growth and tissue repair. Before a cell can divide by mitosis, it must first duplicate all of its contents, including its DNA. This happens during a stage called interphase, which actually takes up most of a cell's life. Interphase is often divided into 3 phases: G1, S, and G2. During G1, the first gap phase, the cell grows and carries out its normal functions. It synthesizes proteins, builds organelles, and accumulates the materials it will need for division. At a certain point in G1, the cell reaches a checkpoint and commits to division. Once past this checkpoint, the cell enters S phase, the synthesis phase. S phase is when DNA replication occurs. The cell must copy its entire genome, all 3 billion base pairs, with extraordinary accuracy. The process begins at multiple points along each chromosome, called origins of replication. At each origin, the DNA double helix is unwound by enzymes called helicases, creating a replication fork. Other enzymes called DNA polymerases then move along each strand, reading the sequence of bases and synthesizing a complementary new strand. By the end of S phase, each chromosome consists of 2 identical copies, called sister chromatids, joined together at a region called the centromere. After S phase comes G2, the second gap phase, during which the cell continues to grow and prepares for mitosis. It synthesizes the proteins needed for cell division and checks that DNA replication has been completed correctly. Finally, the cell enters mitosis, which is divided into several stages based on the behavior of the chromosomes and the cellular structures involved in division. During prophase, the chromosomes condense, becoming visible under a light microscope as X-shaped structures. Each X is actually 2 sister chromatids joined at the centromere. At the same time, the nuclear envelope breaks down, and a structure called the mitotic spindle begins to form. The spindle is made of microtubules, hollow tubes of protein that extend from 2 structures called centrosomes, which move to opposite poles of the cell. In prometaphase, the spindle microtubules attach to the chromosomes at their centromeres, specifically at protein structures called kinetochores. The chromosomes are captured by microtubules from both poles, creating tension as the 2 poles pull in opposite directions. During metaphase, the chromosomes align at the cell's equator, forming what's called the metaphase plate. This alignment is crucial for ensuring that each daughter cell receives exactly 1 copy of each chromosome. The cell doesn't proceed past metaphase until all chromosomes are properly attached to spindle fibers from both poles. When everything is ready, the cell enters anaphase. The sister chromatids are pulled apart, with 1 chromatid from each pair moving toward each pole of the cell. By the end of anaphase, each pole has a complete set of chromosomes, identical to the set in the parent cell. In telophase, the chromosomes begin to decondense, becoming less visible. Nuclear envelopes reform around each set of chromosomes, creating 2 nuclei within 1 cell. The spindle apparatus disassembles. Finally, the cell undergoes cytokinesis, the physical division of the cytoplasm. In animal cells, this happens through a process called cleavage. A ring of protein filaments made of actin and myosin forms around the cell's equator and contracts, pinching the cell in 2 like a drawstring bag being pulled shut. By the end of cytokinesis, what was 1 cell is now 2 cells, each with its own nucleus containing a complete, identical set of chromosomes. The entire process, from one cell to two cells, typically takes about 1 day for a mammalian cell, though this can vary considerably, depending on cell type and conditions. Some cells divide frequently throughout your life. For example, the cells lining your intestines divide every few days. Other cells, like neurons and heart muscle cells, rarely, if ever, divide after you're born. Mitosis is carefully regulated by a variety of checkpoint mechanisms that ensure the process proceeds correctly. If DNA is damaged, if the chromosomes aren't properly attached to the spindle, or if other problems are detected, the cell cycle can be halted, giving the cell time to fix the problem. Or, if the damage is too severe, triggering the cell to undergo programmed cell death, or apoptosis. These safeguards are crucial for preventing cancer, which often arises when cells escape these controls and divide without restraint. The other type of cell division, meiosis, is specialized for producing sex cells, sperm in males and eggs in females. Unlike mitosis, which produces 2 diploid cells, cells with 2 copies of each chromosome, meiosis produces 4 haploid cells, cells with only 1 copy of each chromosome. When a sperm and an egg fuse during fertilization, the resulting zygote has the normal diploid number of chromosomes, with 1 set from each parent. Meiosis involves 2 rounds of division, called meiosis I and meiosis II. Before meiosis begins, during S phase, the DNA is replicated, just as in mitosis. But what happens next is quite different. In meiosis I, the homologous chromosomes, the matched pairs you inherited from your mother and father, come together in a process called synapsis. While they're paired up, they can exchange segments of DNA in a process called crossing over or recombination. This shuffling of genetic material creates new combinations of alleles, different versions of genes, contributing to genetic diversity. After recombination, the homologous pairs are pulled apart and move to opposite poles of the cell. When meiosis Y is complete, there are 2 cells, each with half the number of chromosomes as the original cell, but each chromosome still consists of 2 sister chromatids. Meiosis II is similar to mitosis. The sister chromatids are pulled apart and distributed to daughter cells. When meiosis II is complete, there are 4 haploid cells, each with a unique combination of genetic material. In males, all 4 of these cells develop into sperm. In females, the division is unequal. 1 cell becomes the egg and receives most of the cytoplasm and organelles, while the other 3, called polar bodies, are small and eventually degenerate. Meiosis is crucial for sexual reproduction. It reduces the chromosome number by half, ensuring that when 2 gametes fuse, the resulting offspring has the correct number of chromosomes. And the recombination that occurs during meiosis means that every gamete is genetically unique, contributing to the enormous genetic diversity we see in sexually reproducing species. Cell division, whether by mitosis or meiosis, is a marvel of cellular choreography. But division alone doesn't explain how a single fertilized egg becomes a complex organism with hundreds of different cell types. For that, we need to understand differentiation. Early in development, as we discussed, the fertilized egg divides repeatedly, creating a ball of cells that all look fairly similar. But soon, these cells begin to change. Some elongate and develop long extensions, beginning their journey toward becoming neurons. Others start producing contractile proteins, on their way to becoming muscle cells. Still others develop secretory machinery and will become gland cells, producing hormones or digestive enzymes or mucus. This process, cellular differentiation, is one of the most profound phenomena in biology. All of these different cell types have the same DNA, the same genetic information. Yet, they express different subsets of genes, leading to dramatically different structures and functions. How does a cell know which genes to express? How does a muscle cell become a muscle cell and not a neuron? The answer involves a complex interplay of internal and external signals. During early development, the embryo is not a uniform mass of identical cells. Even at the earliest stages, there are differences in the distribution of certain molecules within the egg cell, gradients of proteins or RNA molecules that will influence the fates of the cells that form in different regions. As cells divide and the embryo grows, cells receive signals from their neighbors and from the larger environment. These signals, often in the form of molecules called morphogens, can activate or repress specific genes. A morphogen might diffuse through the developing tissue, creating a concentration gradient. Cells close to the source of the morphogen receive a high concentration and activate one set of genes, while cells farther away receive a lower concentration and activate a different set. The activation of certain genes can set off cascades of further gene regulation. A protein called a transcription factor, produced in response to an external signal, might bind to DNA and activate a second set of genes, whose products activate a third set, and so on. These cascades can amplify signals and create sharp boundaries between different cell types. As cells differentiate, they also undergo changes in their chromatin structure. As we mentioned earlier, DNA is packaged with proteins called histones, and this packaging can be modified to make certain regions of DNA more or less accessible. In a differentiated cell, genes that are not relevant to that cell type may be packaged tightly and become essentially permanently silenced, while genes essential to the cell's function are kept accessible and active. Differentiation is not always a one-way street. Recent research has shown that under certain conditions, differentiated cells can be reprogrammed, reverting to a more stem-like state, or even changing into a different cell type. The discovery that adult cells can be induced to become pluripotent stem cells by the activation of just 4 specific genes, a finding recognized with a Nobel Prize, has opened up new possibilities for regenerative medicine and has deepened our understanding of the mechanisms that control cell fate. During human development, differentiation occurs in waves. The first major differentiation event is the formation of the 3 germ layers: ectoderm, mesoderm, and endoderm. Cells in each layer then undergo further differentiation, forming more specialized tissues. From the ectoderm, for example, a subset of cells forms the neural tube, which will become the brain and spinal cord. Within the developing brain, cells differentiate into many different types of neurons, each with characteristic shapes, connections, and neurotransmitters, as well as supporting cells called glia. The ectoderm also gives rise to the epidermis, the outer layer of skin, and to structures derived from the epidermis like hair follicles, sweat glands, and the enamel of teeth. From the mesoderm come the muscles, bones, and connective tissues. Cells differentiate into the cardiomyocytes of the heart, the skeletal muscle cells of your biceps and quadriceps, the smooth muscle cells that line your digestive tract and blood vessels. They also become the cells that build and maintain bone, osteoblasts, which secrete the mineral matrix, osteocytes, which maintain mature bone, and osteoclasts, which break down bone and allow it to be remodeled. The endoderm gives rise to the lining of the digestive tract, from the esophagus to the intestines. It also forms the epithelial cells of the lungs, which exchange oxygen and carbon dioxide with the blood, and the secretory cells of glands like the liver and pancreas. By the time a baby is born, it contains over 200 distinct cell types, each specialized for particular functions. The human body is sometimes described as containing around 200 to 215 different types of cells, though recent advances in single-cell analysis are revealing even more diversity, with some estimates now reaching into the thousands when all subtypes are considered. But cells don't act alone. Individual cells, no matter how specialized, are limited in what they can do. The real power emerges when cells work together, organizing into tissues, organs, and organ systems. Let's explore how this organization unfolds. A tissue is a group of similar cells that work together to perform a specific function. In the human body, all tissues can be classified into 4 basic types: epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Epithelial tissue, or epithelium, forms sheets of cells that cover body surfaces and line hollow organs, body cavities, and passageways. The skin's outer layer is epithelium. So is the lining of your mouth, your digestive tract, your respiratory airways, and your blood vessels. Epithelial cells are typically tightly packed together with very little space between them, forming continuous sheets that serve as barriers, controlling what passes from one side to the other. There are several types of epithelium, classified by the shape of the cells and the number of layers. Squamous epithelium consists of flat, scale-like cells. Cuboidal epithelium has cube-shaped cells. Columnar epithelium has tall, column-shaped cells. Epithelium can be a single layer thick, called simple epithelium, or multiple layers thick, called stratified epithelium. The epithelium lining the inside of your cheek, for instance, is stratified squamous epithelium, multiple layers of flat cells that can withstand the abrasion of eating and talking. The lining of the small intestine, by contrast, is simple columnar epithelium, a single layer of tall cells optimized for absorption. Each cell in the intestinal epithelium has a brush border of microvilli, tiny finger-like projections that increase the surface area available for absorbing nutrients. Epithelial tissues often include specialized cells, goblet cells, scattered among the epithelial cells lining the respiratory tract, and parts of the digestive tract produce mucus, which traps particles and pathogens. In the epidermis, melanocytes produce the pigment melanin, which protects against UV radiation. In the respiratory epithelium, ciliated cells have hair-like cilia that beat in coordinated waves to move mucus and trapped particles out of the airways. Connective tissue is the most diverse and abundant tissue type in the body. As its name suggests, connective tissue connects, supports, and binds other tissues and organs. Unlike epithelium, where cells are tightly packed, connective tissue cells are typically scattered within a matrix of extracellular material that they secrete. The nature of this matrix varies widely among different types of connective tissue. In loose connective tissue, which fills spaces between organs and provides padding, the matrix is soft and contains protein fibers, collagen for strength, and elastin for stretchiness, embedded in a gel-like ground substance. In dense connective tissue, like tendons and ligaments, collagen fibers are tightly packed and aligned, providing great tensile strength. Cartilage is a type of connective tissue with a firm but flexible matrix, rich in collagen and other proteins. Cartilage provides support in places like the nose and ears, covers the ends of bones at joints to provide smooth, low-friction surfaces, and cushions vertebrae in the spine. Bone is connective tissue with a mineralized matrix, hard and strong, yet remarkably light. Bone cells, called osteocytes, sit in small spaces within the matrix, surrounded by layers of minerals, primarily calcium phosphate, deposited on collagen fibers. Despite its hardness, bone is living tissue, supplied with blood vessels and nerves, and constantly being remodeled throughout life. Blood, too, is classified as a connective tissue, even though it's liquid. The cells in blood, red blood cells, white blood cells, and platelets, are suspended in a fluid matrix called plasma. Blood's main functions are transport, carrying oxygen, nutrients, hormones, and waste products throughout the body, and defense, with white blood cells attacking invading pathogens. Adipose tissue is connective tissue specialized for storing fat. Adipocytes, fat cells, accumulate large droplets of lipid, which can be broken down when the body needs energy. Adipose tissue also provides insulation and cushioning. Muscle tissue is specialized for contraction, for generating force. There are 3 types of muscle tissue, each with distinct structures and functions. Skeletal muscle is attached to bones and is responsible for voluntary movements. Its cells, called muscle fibers, are long, cylindrical, and multinucleated, formed by the fusion of many individual cells during development. Under a microscope, skeletal muscle has a striped or striated appearance due to the regular arrangement of contractile proteins. Cardiac muscle, found only in the heart, is also striated, but has some important differences. Cardiac muscle cells are shorter and branched, and they're connected to each other by structures called intercalated discs, which allow electrical signals to pass rapidly from cell to cell, coordinating the heart's contractions. Unlike skeletal muscle, which you control consciously, cardiac muscle contracts automatically, regulated by the heart's own pacemaker cells and by signals from the nervous system. Smooth muscle lacks the striations of skeletal and cardiac muscle. It's found in the walls of hollow organs like the stomach, intestines, bladder, and blood vessels. Smooth muscle contracts more slowly than skeletal muscle, but can maintain contractions for longer periods. Like cardiac muscle, it's involuntary, controlled by the autonomic nervous system and by hormones. Nervous tissue makes up the brain, spinal cord, and nerves. It's specialized for communication, for receiving, processing, and transmitting information. Nervous tissue contains 2 main types of cells, neurons and glial cells. Neurons are the signaling cells of the nervous system. A typical neuron has several parts. A cell body, which contains the nucleus and most of the cell's organelles, dendrites, branched extensions that receive signals from other neurons, and an axon, a single long extension that carries signals away from the cell body to other neurons, muscles, or glands. Neurons communicate using electrical and chemical signals. When a neuron is stimulated, an electrical signal, called an action potential, travels down the axon. When the action potential reaches the end of the axon, it triggers the release of chemical messengers, called neurotransmitters, which cross a tiny gap called a synapse and bind to receptors on the next cell, potentially triggering another action potential. Glial cells, or glia, are supporting cells that outnumber neurons in the brain. They provide structural support, supply nutrients to neurons, insulate axons to speed up signal transmission, and help remove debris and pathogens. Different types of glia have different functions. Astrocytes regulate the chemical environment around neurons. Oligodendrocytes produce the insulating myelin sheaths around axons in the central nervous system, and microglia act as immune cells within the brain. These 4 tissue types, epithelial, connective, muscle, and nervous, are the building blocks from which all organs are constructed. An organ is a structure composed of 2 or more tissue types that work together to perform specific functions. Your heart, for instance, contains all 4 tissue types. Cardiac muscle tissue provides the contractile force. Connective tissue forms the valves and the framework that holds everything together. Epithelial tissue lines the heart chambers and blood vessels, and nervous tissue coordinates the heartbeat. Let's consider a few examples of organs to see how tissues collaborate to create complex functions. The stomach is an organ specialized for storing food and beginning the process of digestion. Its wall has multiple layers, each made of specific tissues. The innermost layer is a mucosa, which includes simple columnar epithelium with goblet cells and other specialized cells. Some of these specialized cells secrete hydrochloric acid, creating an extremely acidic environment that helps break down food and kill bacteria. Others secrete pepsinogen, an enzyme precursor that's activated by the acid to begin digesting proteins. Still others produce mucus that protects the stomach lining from being digested by its own secretions. Beneath the epithelium is a layer of loose connective tissue containing blood vessels, nerves, and small glands. Below that is the submucosa, another layer of connective tissue, and then the muscularis, layers of smooth muscle that contract in waves to churn and mix the stomach contents. The outermost layer is the serosa, a thin layer of connective tissue covered with epithelium that reduces friction as the stomach moves against other organs. The lungs are organs specialized for gas exchange, bringing oxygen into the body and removing carbon dioxide. Air enters through the trachea, which is reinforced by C-shaped rings of cartilage that keep it from collapsing. The trachea branches into 2 bronchi, which branch repeatedly into smaller and smaller airways, forming a tree-like structure. The smallest airways, called bronchioles, end in tiny air sacs called alveoli. The alveoli are where gas exchange occurs. Each alveolus is surrounded by a network of capillaries, the smallest blood vessels. The wall of an alveolus is extremely thin, just 1 cell thick, made of simple squamous epithelium. The wall of a capillary is also 1 cell thick. Oxygen diffuses from the air in the alveolus across these 2 thin layers into the blood, where it binds to hemoglobin in red blood cells. Carbon dioxide diffuses in the opposite direction, from the blood into the alveolar air to be exhaled. The lungs also contain connective tissue that provides structure and elasticity, smooth muscle in the bronchioles that can regulate airflow, and nervous tissue that senses conditions in the airways and lungs and sends signals to the brain. The kidneys are organs that filter the blood, removing waste products and excess water and salts, which are excreted as urine. Each kidney contains about 1 million tiny filtering units, called nephrons. Each nephron includes a cluster of capillaries, called a glomerulus, surrounded by a cup-like structure called Bowman's capsule. Blood pressure forces water and small dissolved molecules through the capillary walls into the capsule, forming a filtrate. This filtrate then flows through a long tubule, where useful substances like glucose, amino acids, and most of the water are reabsorbed back into the blood. What remains, now called urine, contains waste products like urea and excess ions. The urine from all the nephrons collects in a central cavity in the kidney and drains through the ureter to the bladder. The kidney is a complex organ containing epithelium in the tubules and capsules, connective tissue providing structure and support, smooth muscle in the walls of blood vessels, and extensive networks of blood vessels to deliver blood for filtration. Specialized cells in the kidney also produce hormones like erythropoietin, which stimulates red blood cell production, and renin, which helps regulate blood pressure. These examples illustrate how organs are more than just collections of cells. They're intricate structures where different tissue types are precisely arranged to accomplish specific functions. But even organs don't work in isolation. They're organized into systems, and it's only at the level of organ systems that we see the full range of functions needed to sustain a complex organism. The human body is typically described as having 11 major organ systems, though different sources sometimes group them slightly differently. Each system has a primary function, but there's extensive interaction and cooperation among systems. The integumentary system consists of the skin and its associated structures, hair, nails, and various glands. The skin is the body's largest organ, covering an area of about 2 square meters in an adult. It provides a protective barrier against physical damage, pathogens, and dehydration. Specialized cells in the epidermis, the outer layer, produce keratin, a tough protein that makes skin water resistant. Melanocytes produce melanin, which protects against UV damage. The skin also contains sensory receptors for touch, pressure, temperature, and pain, and glands that produce sweat to help regulate body temperature. The skeletal system, made up of bones, cartilage, and ligaments, provides structural support and protection for internal organs, enables movement by providing attachment points for muscles, produces blood cells in the red bone marrow, and stores minerals, particularly calcium and phosphorus. The adult human skeleton typically has 206 bones, though the exact number can vary. Bones are connected at joints, where cartilage often provides smooth surfaces and cushioning. The muscular system includes all the skeletal muscles in the body, over 600 of them, which produce movement by contracting and pulling on bones. Muscles also generate heat as a byproduct of contraction, which helps maintain body temperature. The muscular system works closely with the skeletal system. Together, they're sometimes called the musculoskeletal system. The nervous system, consisting of the brain, spinal cord, and nerves, coordinates the body's responses to internal and external stimuli. It's the communication network of the body, receiving sensory information, processing it, and sending out commands to muscles and glands. The nervous system can be divided into the central nervous system, the brain and spinal cord, and the peripheral nervous system, the nerves that extend throughout the body. The brain is an organ of extraordinary complexity, containing roughly 86 billion neurons, each potentially forming thousands of connections with other neurons. Different regions of the brain specialize in different functions. The cerebral cortex is involved in higher cognitive functions, like reasoning, language, and conscious perception. The cerebellum coordinates movement and balance. The brainstem controls vital functions like breathing and heart rate. The endocrine system consists of glands that produce hormones, chemical messengers released into the bloodstream. Hormones travel throughout the body and affect cells that have appropriate receptors. The endocrine system regulates a wide variety of processes, including growth and development, metabolism, mood, reproduction, and the body's response to stress. Major endocrine glands include the pituitary, often called the master gland because it produces hormones that control other glands, the thyroid, which regulates metabolism, the adrenal glands, which produce hormones involved in the stress response, and the pancreas, which produces insulin and glucagon to regulate blood sugar levels. The cardiovascular system, or circulatory system, consists of the heart and blood vessels. Its primary function is transport, delivering oxygen and nutrients to cells throughout the body, and carrying away carbon dioxide and other waste products. The heart is a muscular pump that contracts rhythmically about 100,000 times per day, pushing blood through a vast network of vessels. Arteries carry blood away from the heart, branching into smaller and smaller vessels, and eventually into capillaries, where exchange with tissues occurs. Veins carry blood back to the heart. The cardiovascular system also distributes hormones, helps regulate body temperature by adjusting blood flow to the skin, and plays a role in immune function by transporting white blood cells. The lymphatic systems, closely associated with the cardiovascular system. It consists of a network of vessels that collect excess fluid from the tissues and return it to the bloodstream. Lymph nodes, scattered along the lymphatic vessels, filter the lymph and house white blood cells that defend against pathogens. The lymphatic system also includes the spleen, which filters blood and stores white blood cells, and the thymus, where certain types of white blood cells mature. The respiratory system brings oxygen into the body and removes carbon dioxide. It includes the airways, the nose, pharynx, larynx, trachea, bronchi, and bronchioles, and the lungs themselves. Breathing is controlled both consciously and unconsciously. The diaphragm and intercostal muscles contract to expand the chest cavity, drawing air into the lungs. When these muscles relax, elastic recoil of the lungs pushes air out. The digestive system breaks down food into molecules small enough to be absorbed into the bloodstream and used by the body's cells. It includes the digestive tract, a long tube extending from the mouth to the anus, and associated organs like the salivary glands, liver, gallbladder, and pancreas. Digestion begins in the mouth, where mechanical breakdown by chewing and chemical breakdown by enzymes in saliva start the process. In the stomach, food is mixed with acid and enzymes. In the small intestine, most digestion is completed, and nutrients are absorbed across the intestinal wall into the blood. The large intestine absorbs water and forms waste into feces. The urinary system, or excretory system, removes waste products from the blood and maintains the body's fluid and electrolyte balance. The kidneys filter the blood, producing urine, which flows through the ureters to the bladder for storage. When the bladder is full, urine is expelled through the urethra. By regulating the composition and volume of urine, the kidneys control the levels of various ions in the blood, the blood's pH, and blood pressure. They also produce hormones that stimulate red blood cell production and activate vitamin D. The reproductive system ensures the continuation of the species. In males, it produces sperm and delivers them to the female reproductive tract. In females, it produces eggs, provides an environment for fertilization and fetal development, and produces hormones that regulate the reproductive cycle. The male reproductive system includes the testes, which produce sperm and testosterone, a series of ducts that store and transport sperm, and the penis. The female reproductive system includes the ovaries, which produce eggs and hormones like estrogen and progesterone, the fallopian tubes, where fertilization typically occurs, the uterus, where a fertilized egg implants and a fetus develops, and the vagina. The immune system, sometimes considered part of the lymphatic system, defends the body against pathogens and cancer cells. It includes white blood cells of many different types, each with specialized roles. Neutrophils engulf and destroy bacteria. Lymphocytes produce antibodies and coordinate immune responses. Macrophages consume cellular debris and pathogens. The immune system can distinguish between the body's own cells and foreign substances, a critical ability. When it malfunctions, attacking the body's own tissues, autoimmune diseases result. When it fails to recognize and destroy cancer cells, tumors can develop. These 11 systems, integumentary, skeletal, muscular, nervous, endocrine, cardiovascular, lymphatic, respiratory, digestive, urinary, and reproductive, along with the immune system, work together in a coordinated way to maintain homeostasis, the stable internal environment necessary for life. They constantly adjust their activities in response to changing conditions, compensating for disturbances, and keeping conditions within the narrow ranges compatible with survival. Consider, for example, what happens when you exercise. Your skeletal muscles contract, requiring more oxygen and nutrients, and producing more carbon dioxide and heat. The respiratory system responds by increasing the rate and depth of breathing, bringing more oxygen into the lungs and removing more carbon dioxide. The cardiovascular system increases heart rate and redirects blood flow to the working muscles and skin, delivering more oxygen and nutrients and carrying away waste and excess heat. The endocrine system releases hormones that increase the breakdown of stored energy molecules. The integumentary system produces sweat to dissipate heat. The nervous system coordinates all of these responses. Or consider the regulation of blood sugar. After you eat a meal, glucose from digested carbohydrates enters the bloodstream. The pancreas detects the rising glucose level and releases insulin, a hormone that signals cells to take up glucose. Muscle and liver cells store excess glucose as glycogen. When blood glucose drops, between meals or during exercise, the pancreas releases glucagon, which signals the liver to break down glycogen and release glucose. If glucose drops too low, the adrenal glands release epinephrine, which also stimulates glucose release. The nervous system can sense dangerously low blood sugar and trigger both hormonal responses and behavioral ones, like hunger. These examples illustrate a fundamental principle. The human body is not just a collection of parts, but an integrated whole, a system of systems, each constantly communicating with and adjusting to the others. And all of this complexity, all of these systems and organs and tissues, all of these coordinated processes emerge from the activities of cells, trillions of them. Each cell is a living entity, carrying out the basic processes of life, taking in nutrients, producing energy, building molecules, responding to signals, eliminating waste. But individually, cells are limited. It's only through their organization into tissues, organs, and systems, only through their cooperation and communication, that the full range of functions necessary for human life becomes possible. Think back to where we began, a single fertilized egg cell. From that one cell, through division and differentiation and organization, the entire human body emerges. By the time you were born, you had somewhere around 26 billion cells. As an adult, you have roughly 37 trillion. These cells are not the same cells you were born with. Throughout your life, cells are constantly dying and being replaced. The cells lining your digestive tract are replaced every few days. Red blood cells live about 4 months. Skin cells live a few weeks. Some cells, like most neurons, can last a lifetime, but many cells are continuously renewed. At every moment, millions of cells in your body are dividing. Millions more are differentiating, taking on specialized roles to replace cells that have been lost. Millions are dying, either because they've reached the end of their lifespan or because they've been damaged and must be cleared away. And yet, despite this constant turnover, you remain you. The organization persists. The systems continue to function. The body maintains itself. This is the miracle of cellular organization. Cells are not just the building blocks of the human body. They are the human body, organized into a hierarchy of increasing complexity. Cells form tissues, tissues form organs, organs form systems, and systems form an organism, a living, breathing, thinking, feeling human being. And within each cell, at every moment, an intricate molecular dance is unfolding. DNA is being transcribed into RNA. RNA is being translated into proteins. Proteins are folding into their functional shapes, assembling into complexes, catalyzing reactions, transporting molecules, sending and receiving signals. Mitochondria are producing ATP. The cytoskeleton is shifting, supporting the cell's shape and enabling movement. Membranes are constantly exchanging materials with the environment. The nucleus is regulating which genes are active and which are silent. All of this happens without conscious thought, without effort. You don't have to tell your heart cells to contract, or your pancreatic cells to release insulin, or your neurons to fire. These processes happen automatically, guided by the genetic programs within each cell, and by the signals each cell receives from its environment. It's humbling to contemplate. Right now, as you read these words, as you breathe in and out, as your heart beats and your blood flows and your neurons process information, 37 trillion cells are working in concert, each one carrying out its assigned role, each one contributing to the whole that is you. You are, in a very real sense, a community of cells, a vast cooperative of microscopic living units, organized across multiple scales, from molecules to cells to tissues to organs to systems to the complete organism. And this organization is not static. It's dynamic, constantly adjusting, constantly responding to change, constantly maintaining the conditions necessary for life. From a single cell, an entire human being emerges. From simple molecules, cells build themselves. From cells working together, consciousness itself arises, the ability to think and feel and experience the world. It's one of the most profound mysteries in all of science. How does matter organize itself into life? And how does life organize itself into mind? We don't have complete answers to these questions, but we know the foundation. We know that it starts with cells. We know that cells contain within them the information and the machinery to build and maintain themselves. We know that cells can communicate, cooperate, and organize into structures of stunning complexity. We know that from a single fertilized egg, following the instructions encoded in DNA and responding to signals from the environment, an entire human body can develop. As you drift off to sleep tonight, perhaps you'll think about the cells within you, the neurons in your brain, firing in patterns that give rise to your thoughts and dreams, the muscle cells in your diaphragm, contracting rhythmically to keep you breathing, the heart muscle cells that have been beating since before you were born and will continue until the end of your life, the immune cells patrolling your body, defending against threats, the stem cells in your bone marrow, constantly producing new blood cells, the intestinal cells absorbing nutrients, the skin cells forming a protective barrier against the world. Each of these cells is a universe unto itself, containing the molecular machinery of life. And yet, none of them is you. You are not a single cell. You are not even a single type of cell. You are the emergent property of their collective action, the symphony that arises when 37 trillion instruments play together. This is how cells build the human body, not through conscious design, not through a blueprint imposed from outside, but through the intrinsic properties of life itself, the ability of molecules to organize into cells, of cells to organize into tissues, of tissues to organize into organs, of organs to organize into systems, and of systems to organize into a living, breathing, conscious being. It's a journey that begins anew with each fertilized egg, that has been repeated billions of times over the history of our species, that unfolds within each of us, from one cell to 37 trillion, from simplicity to complexity, from a single point to a complete human being. And now, as we reach the end of our journey tonight, perhaps you find yourself seeing your own body in a new light. Not as a single thing, but as a community. Not as a static structure, but as a dynamic process. Not as separate from nature, but as an expression of nature's deepest principles: organization, cooperation, adaptation, and life. If you found tonight's exploration meaningful, if these quiet revelations about the cellular foundations of human life have brought you a sense of wonder or peace, consider subscribing to Bedtime Biology. Consider sharing this journey with others who might find solace in understanding the intricate beauty of the natural world. And if you have thoughts to share, questions to ask, or topics you'd like us to explore in future episodes, the comments are always open. Until next time, may you rest easy, knowing that within you, at every moment, 37 trillion cells are working in perfect harmony, building and maintaining the extraordinary organism that is you. Sweet dreams.